right, just to recap for you where we left off yesterday, as we were just talking about a cell in general. We introduced the concept of the cell structure. We supported it with this thing called the cell theory. And let's just review the cell theory really briefly. What is one of the tenets of the cell theory? Anything that's considered to be a living organism is comprised of at least one cell. Good. The second initial, the cell is the basic unit of that organism, or the structure of that organism. Very good. What was the third tenet that was added? Cells come from pre-existing cells. To us, it may make sense now, but an historical concept, right? Someone mentioned spontaneous generation yesterday, and so people used to believe that life would just happen, right, spontaneously. But now we know that cells do divide to produce new cells. And then we talked about some uniqueness, I guess, in general about some cells. We said all those cells are going to have some common features that we'll identify today, like a membrane and organelles and etc. There will be moderate differences in structure and size, which provide big differences in function. And so this is kind of where we left off. We said our cells are, by our definition with cell theory, are the smallest living units. And most of them are microscopic, but some can be upwards of a meter long. Okay? Can you think of a cell type that would be a meter long in the body? Some nerve cells are huge in the human. I think the other species that are giant, like giraffes, are notable. Right? They've got feet-long, yard-long nerve cells. But most cell types are tiny and microscopic. You'll need a good microscope to visualize, right, at least part of the cell. So there's a big range. So not all cells are the exact same size. We concluded yesterday that not all cells have the exact same shape, which we translated into concluded to not all cells are going to have the same function. We gave some variation. We said, well, what about the blood cell in its simple state? We said, well, definitely that's a great example. When the structure changes, the function is just, in that case, damaged. What we're going to go through in Chapter 2 is just review in better detail the generic regions of a given cell. And the first important region to identify in a cell is the membrane. I'll call it the plasma membrane. You may call it the cell membrane, referring to the same structure. The membrane that separates the cell from where? What is being separated by the cell's membrane? The extracellular fluid from the intracellular fluid. Very good. So we'll talk about the membrane in structural detail in a minute, but right now we just know that its function just provides a barrier, and it certainly does have a regulatory role as well. It's not like a confined, like concrete wall. It's very much going to regulate what traverses in and out of the cell, like things that we listed yesterday, like oxygen, nutrients, waste, etc. So the membrane is going to be very much involved in this process. Another important area of a cell is the cell's cytoplasm, or we said the what? Cytosol, or I could just say, not intracellular fluid. So intracellular fluid, cytoplasm, cytosol. Technically, cytoplasm, the fluid within the cell, the cyto means, prefix cyto means cell, plasma means fluid. Technically, the cytoplasm includes the cytosol, the solution, the actual liquidy part of the cell. And so it also has cytoplasm or intracellular fluids, an all-inclusive term to describe not just the liquid, but everything else in there. Other organelles, which we'll go through in just a moment, and I'll describe these inclusions as well in just a minute. So it's not just, in other words, cytoplasm is not just a word to describe the liquid. It's all the other stuff, right, if you will, organelles, molecules, etc. And what's the other major area that I mentioned? I think yesterday when I had you draw your little cell, or 2 days ago, what's important to acknowledge in our cell type? The nucleus. And so even though technically the nucleus is an organelle, I like to pull it out. It's really and isolate it as a separate item to talk about because it is nucleus is going to regulate basically everything for that life of that cell. As long as it's present. Sometimes described as like a control center or whatever, but it kind of does that, but it's not all of it. Alright, so what we're going to do is go through and identify these different areas, and I want you to, there should be a spot in your handout to draw a generic cell. Let's just draw another cell like we did the other day. Again, you can draw a cute little round cell like in your handout, or you can draw a red blood cell, nerve cell, sperm cell, egg cell, whatever. But just keep it simple. We're going to draw these three areas. Labeling the plasma membrane, labeling the nucleus, and then labeling the area which is considered to be the cytoplasm. So a little spot on page 1 of your Chapter 2 handout. Label the three different areas: the membrane, the cytoplasm, and the nucleus. You may add additional details by identifying that extracellular fluid as well. So you could say extracellular fluid, or you could be more specific and say interstitial fluid. So those three things listed there, so the plasma membrane, the cytoplasm, and the nucleus. These are the three things that we're going on. So this is just a really oversimplified view of a non-specific looking cell. We're going to go into better detail. We're going to look at the membrane in much better detail. We're going to look at the cytoplasm and the organelles in greater detail. And of course the nuclear structure as well. Alright, so we're going to head to our little cell. The first structure that I want to address is the plasma membrane. In your image that you just drew, and in some of the images that, like in the handout that you're going to turn on Tuesday, the membrane looks plain, right? It's usually just like a single line. Sometimes maybe someone might add 2 lines or something like that. But the membrane is obviously much more complex than a straight line. And so what we're going to do is look at the molecules which comprise the membrane, and obviously look at the function of those structures in the membrane as well. So on the image here on the projector, the far right image is generally how you see a membrane. It looks like a boundary, right? However, if we look at a magnified detail, you can see it's a complex kind of mosaic is a word to describe it, of different molecules. We'll talk about the plasma membrane for the entire cell today. However, when we look at any other membrane-bound organelle, it'll have similar features. And so whether I'm talking about the membrane of the cell, or the membrane of the nucleus, or the membrane of the mitochondria, it'll have the same characteristics. So what are some things, before we move on, what are some things that we can, terms used to describe the membrane, the biological membrane? What are some keywords that you've associated with it before? It's semi-permeable. Very good. What does that mean? It allows some things to traverse it, but not everything. Coin that term in a moment. Very good. What else? What about it makes it semi-permeable? There may be channels. Very good. We all are on a roll, so what are the channels made up of? Very good. the main molecule, the primary, the most abundant molecule in the membrane? Phospholipids. Very good. And in our cells, how are they arranged? They do have a hydrophilic and a hydrophobic region, and the phospholipids are arranged in a what? The whole thing would be a mosaic, but the phospholipids are, you remember the term, I think I heard it, bilayer. Okay, so two layers kind of arranged together. Very good. That was familiar. That's great. So we're going to go through and just repeat that information for this. You guys listed already some important molecules which you'll find in a membrane include phospholipids, which we're going to look at their structure in better detail, a little bit better detail. You already identified that there may be proteins in there. There are definitely other lipids in there. Cholesterol is in our cell membranes. Some proteins are called glycoproteins. What is that? What's a glycoprotein? It has a protein with a sugar. So some proteins have sugars attached to them. There are also glycolipids. What's that? A fat with a sugar group attached to it. The proportion of phospholipids and cholesterol or whatever is going to vary between cell types. So the lipids in the brain membrane are going to be different than the lipids of your red blood cell, which would be different than the lipids of your skin cells. Okay, but they would still have phospholipid protein, etc. within them. We already identified that the phospholipids are arranged in a bilayer. So just look at the word, it's not one, but it's two layers, due to the way that the phospholipids are made. And they're chemically considered to be amphipathic. This word may be new to you, or it may be a word that you've heard after chemistry. Amphipathic, what does that mean? Can you remember? Its character was already described this morning, just a moment ago. Maybe you know about the word infection. It contributes to the permeability, yes. It has what? You're right. It's hydrophilic and hydrophobic. The molecule itself is not just strictly hydrophilic or hydrophobic. It's actually, it has both regions. Because it has both hydrophilic and hydrophobic regions, we could say all that, or we could say it's what? The molecule is amphipathic. Part of it is hydrophilic, part of it is hydrophobic. So for those that are unfamiliar or it's been a while, what does that mean? Hydrophobic. Away from water. Translates to water fearing. So part of the molecule is water fearing. Or the term that I'll use, I may say water insoluble. I may say insoluble. Or we could say what? The other word that was used earlier. Technically it's what? Well, the whole thing would be amphipathic, but one part that is hydrophobic, I could say it's hydrophobic, I could say it's water-fearing, I could say it's water-insoluble. Non-polar. Hydrophobic region is non-polar. What the heck is that? What does that mean? It's non-polar. It's not positive or negative, it's neutral. Okay? So it's neutral. It's electrically neutral. Part of the phospholipid is electrically neutral. Or hydrophobic. Or water insoluble, right? But the other part is hydrophilic, water loving, doesn't have passion for water, but water soluble because it is polar, which means it's partially charged. Part of it's neutral, part of it's partially charged. The part that's partially charged can interact with what type of molecule? Polar molecules, and most famously, water. There's a whole heck of a lot in our bodies of what? Water. It's everywhere. I said all of that, or I could have said the molecule is what? Amphipathic. So let's look at the phospholipid itself to put it into perspective. There's a spot for you in your handouts to draw a phospholipid. This is a simplified view, and to kind of put it in perspective. Phospholipids are described to have a head and a tail. The head region is the polar region. The head region is the part that you would find where? The hydrophilic region facing the fluids, either the extracellular fluid or the intracellular fluid. The tails must be what? Hydrophobic. Does anybody know what kind of molecules you'll find here? What molecules make up the tails? Fatty acids. So basically, carbon-hydrogen chains, hydrocarbon chains. So this is a single phospholipid molecule, kind of oversimplified. We can see the polar head region. That's the region that's going to interact with our fluids, either the intracellular fluid or the extracellular fluid, depending on which leaf it's in the membrane. And the fatty acid tails are just big hydrocarbon chains, which are neutral and therefore nonpolar and then hydrophobic in nature. They would repel one another when placed. They would repel water when placed in a watery environment. What questions do you have about this kind of basic structure of a single phospholipid molecule? There's a little bit better structural detail of a phospholipid. You can see the saccharine tails and the charged head. Any questions at this point? Let's look at the individual phospholipids arranged in a bilayer. Look at that. And again, this is another just simplified view so we can understand it in simple terms so we can build from there. And so we've got multiple phospholipids in one leaf adjacent to another. And notice the arrangement in the layers. How are the phospholipids arranged? So in the bilayer, how are the phospholipids arranged? Very good. On both, within the whole layer, right, both layers, the heads are pointing out where the fluids would be found, and the tails are internalized. So they're not stacked per se. The tails are close inward. Because they're what? Hydrophobic. They're repelling where there's water. Everywhere. Go ahead and draw a simple bilayer in your handout as well. And review the hydrophilic versus the hydrophobic or polar and nonpolar regions in this image as well. on. And you can certainly come back and fill in more if you'd like to on your image. But we're going to go on and look at what other molecules are found in phospholipids and bilayers. It's not just phospholipids. There's other molecules in there as well. This concept, or the better detail or understanding of the membrane, is fairly recently learned. Right? So 1972, this term, the fluid mosaic model, was coined by an article published in Science magazine. Up till now, the real structure of the membrane wasn't really understood. And so the scientist Singer Nicholson described the plasma membrane as we did a moment ago. It's actually not just phospholipids in a bilayer. There's other lipid molecules, and there's also proteins in this structure. And it's in a mosaic pattern. What does that term describe? What is a mosaic describe? Fluid moving, even in an artistic sense. Right? If I go to the art museum and I see a mosaic, what is it? You ever seen a mosaic art something? Art still pretty nice. Okay. I've seen it. How would you describe it? Like small pieces, all looks random, right? There's no pattern to it. And so that's the same kind of characteristic of the membrane. Not just this perfect looking phospholipid bilayer like you tried to draw in your hand on a moment ago. There's other molecules in there, and where it's kind of irregular looking if you were just kind of like a snapshot. There's lots of other molecules interspersed in there. We will add to it that it, because of something in there, it does become actually fluid in its nature. Not static or rigid. If we look a little bit at it, you have a similar image, 215 in your textbook. We can see the mosaic pattern. We showed this image a moment ago. But we can see it's more than phospholipids. The big purple blobs here are representing proteins. The yellow four-ring structures are our cholesterol molecules. You can see some glycolipids, some glycoproteins. These are some things that you'll label, practice labeling in your handout. They'll turn in on Tuesday. Just quick history, which I won't quiz you over. I won't hold you accountable for it. But I just want to show you the history real quick. Back in the late 1800s, it was presumed that there has to be a lipid component to physically separate a cell from its outer liquid environment. But it was believed to just be a single layer of lipid, and the characteristics weren't known. In the early 1900s, it was believed to have a phospholipid, but just a single bilayer. So they're like, well, there's liquid on the outside, so there has to be a polar region. Well, have a phospholipid. We move forward again to the early 1900s, and we can see that scientists have said, well, no, it's not just one. There's water everywhere, so there's actually two layers of phospholipids. It wasn't until approaching 1940s, so like 1930s, it was identified that there are proteins that associate with the membrane. But look at the image. Look how the proteins illustrated in the purple were believed to be associated with the membrane. Look how they were. How were they assumed to be? On the outside, kind of like sandwiching the phospholipids. Then, in the 50s, like 52, 55, like 55, 56 in there, the transmission electron microscope, it was been already invented, but it had become commercially available. Right? So it was becoming to be widely used. And it was identified that, or confirmed, I guess, that it really is a phospholipid bilayer. If you look closely at this image, this is a membrane of a cell. You can see the cytoplasm in the dark staining area. And if you look, can you see that it's described as a trilaminar appearance? So how many layers? 3. The dark would be the phospholipids, phosphate here, and then the white would be where the lipids are located, and the next layer would be the other polar group, phosphate containing group. So it was confirmed, and then it was also identified that the proteins may not only be on the outer membrane at our area, but they may also be integral or integrated into the membrane, traversing. And now, right, we move forward. We now know better detail of the protein. We know the structure in great detail, and we're knowing better than what? What? Organized and setting you up. We know their structure. We know their function a lot better. Very good. So if we look at some proteins that are associated with the cell membrane, some types of proteins, generically speaking, and from the bottom we hand out different types of proteins. One, they may be integral. And so we're just generically just say a protein associated with the membrane. One, it's integral. I-N-T-E-G-R-A-L, integral. Look at that integral membrane protein. How might we, what do we need to know about that protein for it to be integral into the membrane? It's embedded in the membrane. And what about it would allow for it to be embedded in the membrane? What characteristics should it have? It may be a transporter, it may be a channel. It could be a receptor. Good. Chemically speaking, what kind of characters would it express? How can we describe it? A new word maybe to you after today, about 10 minutes ago. Amphipathic. Very good. How do you know that it should be amphipathic? Very good. You notice that our integral membrane protein is going to be found in both the polar and the nonpolar regions, and may even extend into the fluid, where it should certainly be polar. And so these integral membrane proteins should be amphipathic. Now what they may do, maybe transport, receive, whatever the case may be. But if we were to just keep it simple, they've got to be amphipathic because they're traversing at least one, but maybe two leaves of the membrane. Does that make some sense? Are all proteins associated with the membrane integrated into the membrane? Some may be called what? Peripheral. Some proteins are considered to be peripheral membrane proteins. We can see an example here in the cytoplasm. You can see an example facing the extracellular fluid as well. How might you describe a peripheral membrane protein? It's strictly what? Polar. Alright, so we looked at the membrane so far. We said there's phospholipids that are amphipathic, arranged in a bilayer. There's also proteins associated with the membrane. Some are integral, which are also amphipathic. Some are peripheral, which are strictly polar. In addition to some of these proteins, we can see here in this image, some may be really simple, some may be more complex. We'll address those in just a minute. There are also, I'm getting ahead of myself. I'm sorry. So let's look at some functions of those proteins associated with the membrane, and then we'll go on to other molecules in the membrane. Some of them were already highlighted in class today. So we know that proteins are there to do work inside of the membrane. And it was already identified that proteins are there to serve as transporters. And I'm going to draw a little cell on the board. If you want to draw some too, in the little image in your handout. This, like most lists that I'll have this semester, is not all inclusive, but it's a good idea of the diversity of proteins associated with a membrane. Keep in mind, these proteins that we're addressing today are in the membrane. I'm not talking about proteins that are free in the blood or free in the cytoplasm or something like that. Keep leaving them with the membrane, how they might function. I've got a couple of cells here, and we're going to see, just draw some generic looking proteins and molecules and kind of see what they might do. The little pink structure I drew is the membrane, and you can see the two cells separated from one another. Many of our cells have channels that serve to transport molecules. What does that term channel conjure up? Kind of like a tube that's what? Allows things to go through. Like a tube that lets certain things go through. And so some proteins are associated with the membrane, and they are called channels. I'm going to draw my protein here. It's definitely not complex. The blue would be the protein. You can see the opening. It's a channel. Would this be an integral or a peripheral protein? Definitely integral. The only way it's going to form a channel is it's got to get to that hydrophobic region. So sometimes these are described as like barrels. So it's hard to see in the image which I've drawn here. But if you could go back to your phospholipid bilayer, look at all those phospholipids. So a protein has to squeeze in there and push out which region of the membrane. Let me try to push out of the way. What part of the phospholipid? What? Not the head. Well, the head does get out of the way, but really the bulk of it we're trying to get out of the way. What part of it? The what? The tails, the fatty cells, right? So if I have a channel that's pushing out the tails, what kind of molecules think probably would move through a channel? So the tails are now pushed out of the way. Am I doing a good protein? They are pre-A actually. I'm a protein, phospholipids are around me, and I'm opening up. What kind of molecule could go down across the cell membrane? Oxygen actually doesn't need a channel because it's what? It's small and neutral. Does this end up being water? Something like water, which is what? What is it about water? We said it's it is a compound and it is polar. Polar. Okay? Polar molecules are, and maybe ionic, they are definitely peripheral, what do you think? Well, it depends on where, that's just a location, so they're hydrophilic. And the fatty acid layer is hydrophobic. So if I put a channel through, pushing that fat layer away, what type of molecules could go through a channel? Something that's hydrophilic or polar charged. Our transport channels, our channels are usually there to transport polar molecules, charged molecules. Does that make sense? Because otherwise, charged particles would be repelled by which part of the phospholipid bilayer? The hydrophobic. Very good. Repel you. The channels are in there to allow for charged particles, polar particles, to move the transport molecules, and it would be more considered to be a carrier for a molecule instead of just a channel. But what does the term carrier imply? It carries, it attach, it binds. So channel's open, so the molecule would just flow through, and a carrier would bind and carry it through. That's definitely where it's all little. That's supposed to be my carrier. It's not completely open, right? It's going to bind to something and transport it across. The most prominent carrier that we're going to talk about this semester is called the glucose carrier. And so carriers are usually used for carrying molecules that are too big. They may be nonpolar, but they're just physically too big to fit through the membrane. So we've got to carry them. So proteins can be associated with the membrane to transport molecules through examples like channels or carriers. Proteins may be also associated with the cell membrane to aid in this concept of this term called cell-to-cell recognition. What is that? What does that mean? What's the term cell-to-cell recognition kind of conjure up? Two cells interacting with one another. You're probably most familiar with this if you've ever talked about your immune system, or maybe like your blood cell type, like I'm A positive or whatever. Okay. The way that cells talk to some cells talk to one another, is by utilizing proteins, and more specifically glycoproteins. So I'm going to draw an integral membrane protein, and then it's going to have a carbohydrate branch. I'm drawing in red or some green. It's kind of out there, waiting to interact with another cell type. So if I want to draw, this is the third cell in this image. The third cell would also need a what? A protein, a glycoprotein for cell-to-cell recognition. Far from anatomically accurate, but I think hopefully you can get the idea from this image. So those membrane proteins are facing extracellularly. The glycoproteins are facing extracellularly, and they can interact with other cells. That interaction would be another form of cell talking, right? And the immune system say, well, that's okay, or maybe say not okay and attack, or whatever the case may be. The proteins are there for recognition. Okay? So we can find these carbohydrate groups associated with proteins, but also with lipids, and they're described as being fingerprints or characteristics to help identify one cell from another. Because our cells can't verbally talk and say, or show a badge or something like that. That's kind of like showing us badge, like, here's my credit. We'll probably talk a whole heck of a lot, in addition to our transporters, the role of receptors, protein receptors in a cell membrane. Receptors aren't located only on the membrane, but when looking at the membrane is important. So we can have some receptors, some proteins acting as receptors, and what do? They receive. What do they receive? Molecules. If I have insulin in my blood, my cells better have insulin receptors. If I have dopamine in my brain, the cell that's trying to talk to you should have dopamine receptors. So receptors bind with specific molecules It may open or close a channel. All kinds of cool things happen when a receptor receives a specific molecule. Let's draw the line again. Little purple blob I drew up there would be the molecule that binds to it. Yes. Yeah, so I can call it a molecule which is binding. I can call it a ligand, messenger, chemical, hormone, neurotransmitter, whatever. We will go into better detail on that terminology. I'm pretty sure it's chapter 5. But right now, I'm just going to warm you up for it. Ligand is just a generic word without having to get technical about what it is. So a ligand binds to a specific receptor. The ligand may be a hormone. The ligand may be, it could be another protein. It could be, it could be the aspirin which you take for your headache. It binds to a specific receptor. It's just a generic word without getting specific. Does that address your question? Right, so proteins can serve as carriers, transporters, right, channels. They can aid in cell-to-cell recognition. They can serve as receptors. Some other broad categories of proteins associated with the membrane is they may be enzymes. I'll draw up here in green. So next to my receptor, I'm going to draw a little enzyme in the green. The receptor may activate an enzymatic pathway, like we'll learn in chapter 5. Do y'all remember, did you study the CAMP, cyclic AMP pathway? Well, you will soon. I think in the third year. We'll get into that. Chapter 5, I think that's what that is. So, enzymes can be associated with the cell membrane as well. What do enzymes do? They speed up chemical reactions, right? They're described, coin term, biological catalyst, and they regulate specific chemical reactions and enhance them, speed them up. Acting as enzymes, some proteins associated with the membrane may be involved with cell adhesion. I'm going to pull the next two up at the same time because they sound familiar or similar at the same time, at face value, excuse me. So some cell proteins may be involved with processes called adhesion or attachment. It sounds similar, right? Adhesion, the difference is going to be the magnitude of binding and really the duration. So some proteins may be used for adhesion, which is kind of temporary. And our attachment is for long-term, like for forming tissues. Remember we said some cells can be bound together. They're bound together by proteins that allow for them to attach. Conversely, some proteins may just allow for temporary interaction. So for example, if we go back to my proteins which I drew for cell-to-cell interaction, the way that they aggregate together is because there's other proteins that kind of are like a, described as a tether. You use the word tether or dock. They allow for docking. If you've ever been on a boat, you can dock, right? It'd be like a rope pulling the boat to the dock or something like that. There are these proteins that hang out between cells that allow for this docking, if you will, or this adhesion. So the blue and the red are like cell adhesion molecules. The abbreviation in this image, CAM, stands for cell adhesion molecules. The proteins allow for cells to touch temporarily so that they can maybe interact or whatever the case may be. additionally, some proteins may be associated with the membrane that allow for two cells to interact, but it allows for a permanent or more long-term type of interaction between the cells. So for example, in these cells, they will be adhered together. The different types of junctions we'll study in, I think, this chapter actually. So the little red dots that I drew, they're adhering or attaching, our cells together so they don't rip apart. Can you see the red? Let me make them bigger. And even these cells need to be attached to, say, some connective tissue or something like that. And so I'm going to draw some proteins, some fibrous proteins. It's going to look kind of like a mess, but you'll see how the proteins interlace. The strings that I'm going to draw proteins. So from your handout, in your chapter 2 homework, you'll learn about the cytoskeleton. There's proteins inside the cell too. And they attach to the membrane. And then they attach to proteins outside that would be attached to another set of cells. So the green would be proteins, they're extracellular. They allow for the interaction of, I'm just going to draw another cell type on here. So our proteins, the green proteins are the examples of proteins used for attachment, like permanent attachment forming in tissue. It's not as temporary. Can you tell the difference between a cell adhesion molecule versus a protein for a permanent attachment? They're proteins, and they're binding cells together, short-term or long-term. What questions do you have at this point about the kind of snapshot of proteins associated with the cell membrane? If anybody has a camera or a phone, if y'all want to take a picture of this and then post it on D12, we can share it with the class. That'd be useful. We'll get it in the class or whatever. I appreciate that. Actually, is anybody on the Twitter thing? I'm going to try to get more involved on that. If you get hashtag bio24, you can put it out there. Or in D12, whatever you want. I'll share it that way. All right. So we're talking about the plasma membrane. We said there's phospholipids. We looked at their character. We also looked at the proteins, the integral versus peripheral, and some general regions or general functions of proteins. Where I was headed earlier is there are other molecules in the cell membrane, including a molecule called cholesterol and other sterols and different types of stuff. But cholesterol is an important molecule in the cell membrane. You know, cholesterol is one of those things that gets a bad rap in the media, right? That's what we hear about it in the society, right? In the media, like in the ads on TV, lower your cholesterol, all this stuff. Because just like everything else we do, we tend to overdo stuff, and we overeat bad foods. But cholesterol is critical for membrane construction. Without cholesterol, membranes wouldn't be built right, and they wouldn't work. They wouldn't work right. Okay? And so the question is, well, then what does cholesterol do? It is amphipathic. We're okay with that word now, hopefully. And it gets in there in between those little phospholipids to contribute to the mosaic pattern of the membrane. And it's complex. I teach cell biology also, and so we look at the mechanics of it. And even when I talk my way through it, I still confuse myself on it because of the technicalities. It's very interesting how cholesterol works. So what it does is helps keep the membrane fluid. So it's the presence of cholesterol that prevents the membrane from seizing up, basically, in cold or melting in hot temperatures. So it keeps the in a range of temperatures. This is what gets, I guess, amazes me about cholesterol. It protects the cell on both ends of the temperature spectrum. In hot, it keeps the membrane from just oozing. And in cold, it keeps it from solidifying. One molecule, two ends of the temperature spectrum. Yes?
So when you say that about this cholesterol, are you talking about both cholesterol, HDL and LDL?
When you see the media, we're talking about high density and low density lipids, that is when it's in a vesicle circulating through your blood, which this could certainly go into the blood. There's other brain tissue block or something like that. But there's little membrane vesicles or packets or spheres, and inside of there, chock full of different types of lipids. And it may be cholesterol. The high density, low density, does the saturation of those, maybe the degree of saturation. I can't fully address your question. I do know the media, though, it's a completely different structure in the blood. I'm going to draw it over here if you're curious. That's a membrane, but it's tiny, and it has chock full different types of little lipids packed into it. This membrane has its own little proteins and lipids and etc. High density versus low density tells where it goes. Like green things are other proteins. It's the low density that's going bad, right?
Yeah.
The low density targets cells. It binds with cells in your vascular system. The high density targets other tissues and gets incorporated into the cell and used by other cells, like say your muscle cells or your fat cells or whatever. So what makes it good or bad is this protein. It's a good example of a protein cell-to-cell kind of interaction. And so the high density, these receptors target heart and blood vessels and then dump those there, which is not good. The composition of the lipids in there varies. It's the receptors in that vesicle where it knows if it's good or bad. And so it may be other phospholipids, could be triglycerides, maybe cholesterol as well. It may not be just cholesterol. So I think it's going to depend on your diet and other factors to say what lipid is in that vessel. Yes, and so just your health. So I don't think they're all exactly the same is what I'm trying to say. But the difference is where that's a good setup for protein-to-protein interaction. Where it could dock at the heart or some other tissue that would be caused. Right. So good question. Additional questions? All right. So cholesterol is in a membrane, and what is its job? Keep the membrane is described as fluid. It keeps it from melting in extreme warm temperatures and gelling in cool temperatures. We can manufacture cholesterol, which we'll look at with organelles involved with that. Everybody knows? Which organelle helps manufacture our lipids? Smoothie yard, very good. All right. So we've talked about the membrane, we talked about the phospholipids, we've talked about the proteins, and we briefly mentioned the role of cholesterol. The other major area of a cell is called the nucleus. The nucleus, come back to you. The nucleus, you at least know it contains our DNA. You fundamentally, hopefully, at least know that. That's where our DNA is found. You may not know that some cells may not have just one nucleus. Some cells may actually have multiple nuclei in the mature form. As we develop, some cells fuse together. You become, like, say, 2 or 3 cells join together to make one giant cell. And so those nuclei are retained in those cell types. some cells even are anucleated. What does that mean? Some cells don't even have a nucleus, which doesn't make sense, does it? Because we're characteristically, we said what type of organisms? Eukaryotic. But some cells in the mature state do not have a nucleus, which is our what? What cells in our body do not have a nucleus that are part of our body? Fat cells do, and our red blood cells do not. Red blood cells don't. They kick out the nucleus. Chapter 15 we'll talk about briefly. We'll review that after this. They used to have a nucleus. They're still eukaryotic. Take it out. So red blood cell doesn't have a nucleus. What can it not do? It cannot undergo mitosis. It can't divide. Can't make new proteins. Very good. I just want to point out some important features of the nucleus. One, the nucleus is membrane bound, so technically it's an organelle. So it has a phospholipid bilayer, like we just described, but it's not one, it has two layers. So the envelope is a term to describe those two bilayers around the nucleus. So the nucleus actually has two, and if you look closely, and I'll show another image, you can see one, two, you see the two layers of the nuclear envelope. In this image, the dark purple is the nucleus, so this thing has been pulled out from the cell. It's not the entire cell, it's been pulled out. You can see the fluid inside of the nucleus, and look at it closely at its perimeter. You might be able to discern two phospholipid bilayers. It's two layers. The nuclear envelope has two layers. It also has pores, which are shown here in this image in the yellow. What type of molecules do you think make up the pores? Proteins. Very good. So it's double layered. The envelope is double layered. It has pores. And it also has what on it? Anybody know what these little dots are? Ribosomes. It also has some ribosomes attached to it. So in other words, the nuclear envelope structure is complex because the nucleus is kind of very important for a cell. So it's got to be built well to protect. Another major region of a nucleus is a region inside of the nucleus called the nucleoli. Or a single one would be a nucleolus. US would be singular. Suffix I would be plural. There may be one, there may be two. Nucleoli. The nucleolus is just a region, just a concentration of molecules. So they look dark under a microscope. It's just a concentration of molecules that do what? What are these molecules in the nucleolus usually do? They manufacture our ribosomes. Very good. They give the genetic component of the ribosome. The nucleolus helps build ribosomes. Part of them, not all of them. A third important region of a given nucleus is the molecules that are collected what's called the chromosome. What is the chromosome? Is our DNA with what? Proteins. I thought I heard you say. It's our DNA package, basically, in the histone proteins. So chromatin is just this kind of partially packaged, condensed DNA, as illustrated by the purple fibers in this image. So our chromatin is in there. Our DNA, we know that our DNA is in there, but it's not just DNA
Negative.
The nucleus has a very complex nuclear envelope comprised of two membranes with permeable pores and studded with ribosomes. Another important region of the nucleus is the area called the nucleolus, which is important in the production of ribosomes. Thirdly, maybe most famously, is the chromatin that's compartmentalized there inside of the nucleus. I've got a few images to kind of help better illustrate these regions. This next image is showing the double membrane nature. Kind of the purple bit is the phospholipid bilayer, and you can see how it doubles. There's two of them adjacent to one another. You can also begin to see the pore complex. You can see it acts as a what? Kind of like a channel. We certainly need to get products in and out, but we want to regulate it. And you can also see the outer surface of proteins, ribosomes attaching to them. Here's a freeze fracture scanning image of a nucleus, and we can see very clearly the pore complexes. Can you see the big divots? They're huge. They're big protein complexes, and then tiny little bits associated. What are the little things? Ribosomes. So just a real image to support the cartoonized image. These are real. They're real real. And the chromatin, here's an alternative image of the chromatin, the string thread looking. It's not just purified DNA. It's these proteins called histone proteins that kind of condense it, or do condense it, to keep it organized. What questions do you have about the structure of the nucleus before we look at the components of the cytoplasm? Let's review the cytoplasm. And we identified it's the area basically between the cell's membrane and the nucleus. It's everything else. That's not the nucleus. That's not in the membrane. We identified as well structures that you'll find in there. The true liquid of the cell, which is called the cytosol, or the solution of the cell. We have liquid in there. And we said it's not just a liquidy environment. There's organelles in there, which I was hoping to get through today, which we are not going to get through today, but on Monday we'll run through those. They're going to have a specific design for a specific function, and we'll come back to those. And there's also different types of inclusions in a given cell type. I have the in parentheses the cell type. The type of inclusion varies depending upon the cell type. So for example, my adipocytes probably have what type of inclusion? What is an adipocyte? What is that? Fat cell. Fat cells have inclusions of fat. Okay. My skin cells can store what? Mine not so much. And some of you guys are better than me. Storing what? Pigmentation. Okay. So that varies. My liver can store what? Well, my liver cells can store what kind of inclusions in them? Nutrients. Do you know the name of the nutrient? What? Glycogen. Good job. We're on it today. So you wouldn't see your liver cells storing pigmentation, or your skin cells storing glycogen, but they both have the ability to store something. So what they store depends on what they're designed to do. And of course, we could list for 3 days straight what you would find inside of. I said we weren't going to get through them all, but we've got 5 minutes. We're going to get introduced to them. So to wrap up quite yet, we'll need every millisecond in this class. I want to talk about the organelles that you'll find within a cell. Hopefully this is a review, but I know that it takes multiple exposures to really understand how some of these structures and organelles function inside of the cell. By my definition, I'm going to define an organelle as a membrane-bound structure. That is debatable. Some people don't like to limit it with the membrane. That's fine. There's going to be a whole lot more to talk about. And these membrane-bound structures are the ones that we're really going to focus on this semester. So we'll talk about the structures that have a phospholipid bilayer around them. We'll first talk about the endoplasmic reticulum, which you can abbreviate And hopefully know that there are two subtypes of endoplasmic reticuli. There's a smooth ER and the rough ER. There's another organelle called the Golgi apparatus. We may call it the Golgi complex, Golgi body. Other structures including lysosomes and peroxisomes. And we'll conclude our discussion of the mitochondria in our organelles, because that'll set us up for chapter 3, which is metabolism. Mitochondria. So let's first look at the endoplasmic reticuli. We'll first look at the smooth ER. It was already mentioned what it does. What does a smooth ER do? Not the smooth, lipid synthesis. The smooth ER is very much involved with lipid synthesis like our cholesterol, that we mentioned a moment ago, or our phospholipids that we've been talking about. It does. Smooth ER, we are going to talk about in its role in calcium storage a whole heck of a lot for the end of the semester in our muscles. So one important job of the smooth ER is to manufacture lipids, or steroids or phospholipids, etc. With their origins, here in the smooth ER. My presentation is kind of messed up a little bit. The third and the fourth bullet go together. The smooth ER is modified in some cells so that it does store calcium cations. The smooth ER gets its name because under a microscope it looks smooth. And what that means is it doesn't have these ribosomes attached to it, like we saw in the nucleus a moment ago. And so if we look at a similar image that we had earlier, I can see again the purple double nuclear envelope. I can see a membrane here, but I see something on it, so that can't be the smooth ER, right? And if I look back here on the back, I can see some tubes adjacent to the nucleus. They're tubes. There are no ribosomes attached to it, so that is going to be the smooth ER. This is hollow, so it has a what? What term did we describe those? As a lumen. It's not devoid. It's empty, like it might look in this image. What's in there? What's inside of a smooth ER? There may be proteins. There's definitely calcium. What else? And there are proteins, enzymes working in there. There's definitely fluid in there. And other molecules, right? What else is added? Calcium. So don't think of it as empty. There are chemical reactions happening inside there. Manufacturing lipids, storing calcium. We're going to conclude our discussion here today. We'll pick up with the rough ER. Please review this over the weekend in good detail so that on Monday